everybody. I hope everybody can hear me well. My name is Dirk Löffert. I'm Director Research and Development at QIAGEN, and I'm going to present today some critical success factors in the development and implementation of biomarkers in drug development. To give you a brief overview on what the presentation is about, I prepared the slides. So I will first talk about the impact of biomarkers in discovery and use, how biomarker programs can benefit drug development procedures and processes, what are potential technological variabilities in biomarker research programs and how they can compromise the outcome, but also solutions to this. And in this section, I will introduce a couple of new technology developments which can overcome variability in biomarker assessment. We'll conclude my presentation then with some words on standardization of complete assay workflows, introducing a brief case study, and summarizing in the final slide the presentation. Let me start with this first slide showing the population chart as it's expected to be for Germany in the year of 2050. And what you can easily deduce from this chart is that about half of Germany's population in 2050 will be 50 years old and older. And this implies that many age-related diseases such as Alzheimer, Parkinson, cancer, diabetes are much more prevalent among the population. Thus, there's still a big impact and profitability of the pharma market on healthcare in the future. So very promising market segment. However, when looking at R&D spending versus new products coming out of pharma pipeline, there appears to be a big discrepancy between the amount of money spent and actually new targets coming to the market. And the FDA states that only 14% of the newly approved drugs are considered a significant improvement over existing products, such being under great competition with already available solutions on the market. So questions are, how can new technological approaches benefit new development programs? And that becomes even more striking if you look at this chart, since in the past years, major adverse drug reactions have affected drug recalls from the market, not only destroying value and pipeline value from pharmaceutical companies, but also destroying major shareholder value. And the question, of course, arises, how can this prevent it in future? This has led to major new trends, namely the learn and confirm model, using biomarkers as early as possible in drug development procedures, and to use biomarkers to learn more about on the mechanism of a disease, as well as the drug function on the disease. So major pharmaceutical companies now introduce biomarkers as major selection tools for the drug development procedure already during the R&D phase, drug discovery phase, and usually most compounds need to be backed up by appropriate data through biomarker investigation to proceed to next phases in the development procedure. But to give also a positive example, biomarkers do not only help in eliminating drug compounds from the development process, but also help to rescue those drugs which may have previously abandoned, as is shown in this example, where just the stratification of a certain patient population has helped to rescue the drug for a specific treatment for this patient population being safe and effective. This is just a summary of well-known biomarkers coming from the pharmacogenomics area, which are often used in accordance with the therapy. And two very prominent examples among those are, for example, QIAGEN or also Herceptin for breast cancer treatment. And I will relate to this specific biomarker, the HER2 Neu receptor, later on in my presentation. So the advantage and benefit of biomarkers is quite obvious. It's a tool to save costs by achieving faster go or no-go decisions for certain compounds in the drug development process. helps eliminating compounds early in development, for example, because of their toxicity, but may also help to rescue compounds from failure through better stratification of patients and thereby overall reducing time and resources, saving money in the development process. With the appropriate data backed up in the approval procedure, they can also help to get faster regulatory approval and definitely will benefit to generate safer drugs. On one hand, by better understanding of the functional mechanism of the disease itself, and by the help of biomarkers to elucidate the mechanistic function of the drug compound. And more related to the pharmacogenomic area, better patient stratification is expected to benefit safer drug developments by addressing specific patient populations according to their individual genotype with the appropriate drug and drug dosage. However, it has been well described that often biomarkers also fail to benefit development programs, and there may be two major reasons. One is the biological variability, such as the inter-individual variability, but the other one, which is definitely also of greatest importance, is the variability which may be introduced by the methods or technologies involved. And I would like to focus on this aspect and also introduce new technologies which help to reduce overall variabilities in biomarker programs. This gives a schematic overview on pre-analytical steps and analytical steps involved in an experimental workflow, and describes in which stages variability can affect results of biomarker programs, which can be on one hand in sample collection and storage, the isolation of biomolecules itself, as well as in assaying biomolecules. And I will discuss all these aspects in a little bit more detail on the following slides. Talking first about sample collection and storage, it's necessary to ensure that biomolecules which are the targets are not degraded during the collection procedure and over storage time, and that also artificial gene expression is not induced upon handling and storage of the samples. This is shown in the schematic drawing, where the red line represents the in vivo transcript level at the time point of taking the sample, and what can happen upon storage of the sample, namely artificial induction of gene expression or actually enzymatic degradation and or downregulation of gene expression. So that makes pretty clear that RNA stabilization is a major need to stabilize the biomolecules of interest. And there are two major technologies currently available. One is the PAXgene blood RNA technology for stabilizing transcripts in whole blood samples. The other one is the new all-protect tissue technology, which not only stabilizes transcripts, but also DNA and protein from whole tissue samples. And I would like to show some data on these. First, for the PAXgene RNA blood system. In this case, whole blood samples were either collected with a common EDTA whole blood tube or with a PAXgene blood vacuum system, which was co-developed with Becton Dickinson in a joint venture called Pre-Analytics. And in this case, several inflammation biomarkers were assessed for their stability of the transcript level over different storage times, starting at time 0, of course, going from 8 hours up to 5 days. And as you can see, for one particular inflammation marker, in this case interleukin 8, without preservation of the transcript profile, you see a significant change in the transcript level, which is, however, eliminated by using the PAXgene blood RNA system to stabilize the transcript profile up on drawing of the blood sample, as holds true also for all other different inflammation markers in this case, which show major different upregulation or downregulation in non-stabilized samples. This has been also shown in one study by Müller et al., published in Leukemia, using the ratio of PCR-ABL to ABL as a biomarker for leukemia therapy monitoring. And what this group has investigated is the stability of the biomarker, comparing the sample which was freshly taken, stored for only 2 hours, and comparing the transcript level with a sample which was stored for 72 hours. using either, again, a stabilization reagent like PAXgene blood RNA or an unpreserved blood sample. And as you can see with the preserved RNA sample, the ratio of the fresh sample, of the transcript from the fresh sample with the stored sample maintains pretty much the same, leading to very high correlation of the data points, whereas in unpreserved blood sample, this correlation is no longer given, showing clear degradation of the message over time of storage, and thereby diminishing the accuracy of the results from this biomarker analysis. We have recently introduced a new stabilization reagent, which not only stabilizes RNA, but also DNA, RNA, and proteins, and in this case from tissue samples, which was made possible by the use of the so-called all-protector technology, which are small molecules with affinity to biomolecules infiltrating the tissue sample, and in principle covering the biomolecules and thereby making them not longer accessible by, for example, nucleases. And these complexes are broken up during the tissue lysis procedure, also destroying the all-protector molecules, which can be therefore very efficiently removed from the sample, leaving behind intact biomolecules like RNA and proteins. And the protecting effect of this new technology is shown on the next slide in two representative examples. On the left side of the slide, you see the stabilization of the C-Phos transcript level using either the new all-protect technology or an unpreserved sample. And as you can see, with the all-protect technology, the transcript level of C-Phos stays the same, whereas in unpreserved sample, the gene expression of this particular transcript can vary a lot over storage time and goes along with high variability. But the same holds true for proteins. So proteins can be also now preserved in tissue samples. And in this example, we have compared the activity of acidic phosphatase, benchmarking the all-protect reagent against the liquid nitrogen stored sample, and looking either at a sample which was stored at room temperature for up to 1 day or refrigerated up to 7 days, utilizing similar protein amounts and seeing similar activity of the proteins from the different samples compared with a liquid nitrogen stored sample. So showing pretty much high stabilization, not only of RNA, but also of proteins in tissue samples. The next source of variability, which I would like to point out, is derived from the isolation procedures of biomolecules itself, as well as from the sample complexity. So sample complexity means, for example, in proteomic workflows, that low abundant proteins may be masked by the presence of highly abundant proteins, such as serum albumin. And there are two strategies how to increase sensitivity and reliability of such procedures in proteomic workflows, namely by either using a depletion procedure or an enrichment procedure. Depletion can comprise, for example, depletion of high abundant proteins such as serum albumin using the Qproteome human or murine depletion kits, but could also mean a specific enrichment of proteins according to their cellular localization, for example, nucleus localized proteins or plasma membrane localized proteins, or selective enrichment of proteins according to their post-translational modifications, such as phosphorylation, so to in particular and specifically enrich proteins of interest for further downstream analysis. This shows one example utilizing the Qproteome Phosphoprotein Purification technology to reveal the presence of low abundant phosphoproteins from one and the same sample, comparing a non-enriched sample with the enriched sample and analyzing similar amounts on a 2D shell electrophoresis, showing that low abundant phosphorylated proteins can be still detected with this purification and enrichment technology, which would be otherwise not detected in a non-enriched situation. Another example which I would like to draw your attention to is the analysis of proteins from very difficult samples such as formalin, fixed, paraffin, embedded tissue samples. In this case, proteins are of course cross-linked by the presence of formaldehyde, which makes it very difficult to, on one hand, retrieve them from the tissue sample, on the other hand, to analyze them. And I would like to show this new technology with one biomarker example, namely the detection of the HER2 receptor protein, as is necessary for the administration of the Herceptin therapy. Usually, as an introductory stage, samples are investigated by immunohistochemistry analysis for the presence of the HER2 receptor, and a score of 0 or 1 plus is usually not indicated as suitable for Herceptin therapy. A score of 3 plus is a clear indicator that such a patient may undergo a Herceptin therapy, whereas a score of 2 plus somehow gives equivocal results, and a further selective screening is necessary, like a fluorescent in situ hybridization for the HER2 gene, where the question is whether this gene has been amplified, because in this case, those patients would be also feasible to undergo a Herceptin therapy. With the accessibility of proteins directly out of FFPE samples, there's now another possibility for protein-based biomarker analysis through the Qproteome FFPE kit. In this case, you can take unstained tissue section and isolate with this technology proteins for further downstream analysis, such as protein lysate arrays or Western blot analysis. In this case, from this sample, which was scored as type 2 positive by the group from the university in Munich, they could see from the positive sample clear presence of the HER2 gene as indicated by staining with the HER2 antibody, and could also reveal the full-length protein of the HER2 receptor on Western analysis, and to be able to semi-quantitatively even estimate the amount of this protein compared to an endogenous normalization protein. The big advantage of such a technology is now that you do not only indirectly assess the presence of your biomarker, but that you can actually detect the drug target directly from your protein sample in this case. Another problem which can arise when using patient samples for biomarker analysis is the quite limited amount for genomic analysis as well as RNA analysis. And for this slide, I would like to introduce technology which can be employed to immortalize genomic DNA samples and to generate, in principle, unlimited resources for genotyping analysis, being it actually directly used in the biomarker discovery program or later on in a retrospective study. And this technology works with really very small starting DNA amounts. One can start with purified genomic DNA, for example, from blood spots or buccal swabs or mouthwash, or one can also actually use directly blood samples like half a microliter of blood, which is really sufficient to generate later on up to 40 microgram of genomic DNA. The procedure starts with a cell lysis or alkaline denaturation of the starting DNA template. After a neutralization step, the DNA is mixed with a so-called REPLI mix, which comprises all necessary reagents like the phe29 polymerase, nucleotides, and random oligomers, and then the reaction is simply conducted for several hours at isothermal temperature of 30 degrees centigrade and yields, depending on the format and time, up to 10 microgram or 40 microgram of identical genomic DNA, representing all the genomic loci present in the starting material. And this material is then directly suitable for genotyping analysis, such as SCR genotyping or SNP genotyping based on TaqMan technology. This slide gives a more detailed representation of how this so-called multiple displacement amplification technology works. And this technology is unique to certain polymerases such as phe29 polymerase. So you start with the alkaline denaturation, then you hybridize random hexamer primers to the template, which ensures that all genomic loci are covered during the amplification reaction and are equally represented in the amplified material afterwards. phe29 polymerase attaches to the random primers and extends the primers. And now a very specific feature of Phe29 polymerase comes into the play, so to speak. In contrast to example TAC polymerase, this enzyme does not hydrolyze target strands sitting in front of the enzyme, but rather displaces the template strands from the DNA template. This again generates a single-stranded DNA template, which is now again able to bind to random oligomers. And these random oligomers, of course, can be extended again by Phe29 polymerase. By this mechanism, generating in principle a hyper-exponential amplification procedure, yielding extremely high amounts of DNA while keeping the representation of all genomic loci. This is achieved by the very special features of Phe29 polymerase. In contrast to TAC polymerase, which is rather a DNA repair enzyme involved in the replication process, Phe29 polymerase is an actual replication polymerase. So it has extremely high processivity and generates amplification products up to 100 kilobases without dissociating from the template strand. And this ensures that all genomic sequences are actually copied without any bias in the representation of the genomic DNA. And I will show this on the next slide. Another specific feature of this polymerase is its intrinsic high proofreading activity, which ensures highly accurate copying of the template strand without introducing major additional mutations into the target DNA strand. This slide shows a comparison of 3 different whole genome amplification methods to enable unlimited PCR and genotyping analysis from your starting DNA. One, the as described multiple displacement amplification technology, or 2 PCR-based whole genome amplification procedures. In this study, 8 different SNP loci were investigated using TaqMan technology and were checked whether they are equally present in the amplified material compared to the non-amplified starting material, using different starting DNA amounts coming from 300 nanogram going down to 300 picogram. And as you can see from this slide, the multiple strand displacement amplification technology preserves the genomic representation of all 8 different loci throughout different ranges of DNA amounts, whereas the PCR-based amplification approaches show highly variable results with an increased risk to lose certain genomic loci due to dropout during the amplification procedure, leading in such instances, of course, to incorrect data and genotype interpretation. So I think this nicely shows that the unique technology based on Phe29 polymerase preserves the genomic loci representation very well. And this very same technology, now that's a very new and recent development coming to the market only end of this month, has been also applied to limited RNA samples as they can occur if you want to do gene expression analysis, for example, real-time PCR-based starting from a few cells, or again from previous sample material such as patient material, fine needle biopsies. And in many of these cases, you just have too few RNA amounts to be able to run and analyze all the different transcripts you're interested in, or to repeat certain analysis later on. So the problem is, how can this be overcome? And the current more or less home-brew solution is pre-amplification of cDNA by using PCR primers, which are then also subsequently used in the real-time PCR analysis. However, this procedure has the limitation that you can analyze only those transcripts which you already pre-amplify from your cDNA sample. So you cannot start later on taking the same cDNA sample and analyze other transcripts. A further limitation exists due to the high presence of primers which need to be included in such a pre-amplification reaction. This may limit the efficiency and sensitivity for some of the transcripts you are going to investigate. With a relatively simple trick, we have now being able to adopt the Phe29 polymerase technology also for RNA amplification or cDNA amplification, mainly by ligating the resulting cDNA targets after the reverse transcription reaction together, generating a long enough target molecule for the Phe29 molecule polymerase to efficiently and representatively amplify the cDNA. And this procedure is very straightforward, requires only minor amounts of RNA, such as in the low nanogram range. And let's say from 10 nanogram, after 2 hours of amplification, you end up with 10 microgram of cDNA. After 8 hours of amplification, up to 40 microgram of cDNA, starting with just a few nanogram of total RNA. And this technology, like for whole genome amplification, preserves very well, in this case, also the transcript profile, leading to no bias in your transcript analysis, as is shown in this example, where we have analyzed 50 different transcripts, comparing the relative transcript abundance from a non-amplified cDNA and from the cDNA which has been amplified with a new quantitative whole transcriptome kit. And as you can see from this chart, there's a very close correlation of the gene expression levels for the various transcripts coming from either the non-amplified or the amplified cDNA sample, showing a high preservation of the transcript profile. Another well-known feature, which is due to most RNA amplification procedure, is the observed bias of RNA sequences which are more located towards the 3' region of the transcript. But since with this new technology we make use of random primers which cover not only all transcript but also all transcript regions, we do no longer observe with this unique amplification technology any bias with regard to position effects on individual transcripts, as is shown on this example, where we have amplified either PCR products located to the 5' region of the transcript or the 3' region of the transcript. And as one would expect, if there's no bias introduced during the amplification reaction, the CT value for both regions appear to be pretty much the same. With this example, I would conclude the portion or discussion on the pre-analytical steps and would like to come now to variability and new technology solutions, how to overcome variabilities in the analytical steps, mainly as it's related to real-time PCR-based gene expression analysis, covering aspects like reproducibility, specificity, and quantitative range on different detection platforms. And one very appropriate technology to further increase reliability of gene expression data is the use of the so-called multiplex real-time PCR technology. Why is this the case? So multiplex real-time PCR allows you to co-amplify in the same reaction not only your internal reference gene like GAPDH or 18S ribosomal RNA, but also your target gene of interest or even several target genes of interest. And since you do not add variability due to additional pipetting steps or different position effects on your real-time PCR plate in the instrument, you can yield much more reliable quantitative results by co-amplifying the transcripts of interest in this one and the same reaction. This also helps to further conserve precious sample material, since you can simply analyze more transcripts from the same starting amount. You can increase throughput and thereby also reduce reagent costs, since you can generate more data points out of a single reaction. And since all our reagents were optimized to work with all commercially available cycler platforms, you gain already some standardization throughout different labs if there are different real-time PCR instruments to be used. However, I have to say that duplex or multiplex real-time PCR is not a very trivial method, and it has been well published in the literature that it suffers from lower sensitivity and that gene expression levels estimated based on multiplex real-time PCR may differ significantly between singleplex results and duplex results. And there are a few very obvious reasons to this. One is the potential interference of the different primers and probes in one and the same reaction. generating non-specific PCR products which compete for the desired PCR products later on in your PCR reaction, and diminishing in principle overall sensitivity of your assays. Another one is that even if you design your primer and probe pairs with the same algorithm, like let's say Primer Express software, you will experience that hybridization kinetics of the different primers and probes may be quite different. And if there are primers and probes contained in your multiplex reaction which hybridize with a lower efficiency, this will also lead to a lower PCR efficiency of this particular target in your amplification reaction. And another reason for problems in multiplex real-time PCR is the well-known fact that more abundant transcripts become preferentially amplified during the amplification reaction and therefore do not yield reliable gene expression results. We have introduced two new technologies to overcome these problems, and I would like to briefly introduce them on the next slide. One important factor is a very stringent hot start procedure, as can be, for example, achieved with a chemically modified hot start tag polymerase, which ensures that all enzyme activity is not present until the initial reactivation step at the start of the PCR reaction. This ensures that no premature non-specific amplification products are generated, which can later on compete for the yield of the desired PCR products, and thereby affecting those PCR efficiencies. However, another very important factor is the PCR buffer formulation itself. On one hand, one can achieve much higher PCR specificity by appropriate cation formulations, in principle destabilizing by the presence of ammonium ions non-specifically bound primers, and thereby improving overall PCR specificity in each cycle during each annealing step. But probably the most critical factor which we introduced in order to enable multiplex real-time PCR is the inclusion of so-called macromolecular crowders. Macromolecular crowders are well known from very old molecular biology principles and experiments, such as ligation experiments, where macromolecular crowders such as polyethylene glycol are used to direct inter- or intramolecular ligation events. And in this case, we have made use of macromolecular crowders in order to improve hybridization kinetics of those primer and probe pairs which hybridize suboptimal, and thereby we can equalize the overall hybridization kinetics of the different primer and probe pairs in the multiplex PCR reaction, and yielding thereby a similar PCR efficiency, which is substantial for accurate gene expression results. And this new reagent technology has been actually the topic of several publications, and two more recent publications I would like to introduce here. One has been published in Analytical Biochemistry by Novartis Pharma from a group in Basel in Switzerland, and another example on which I would like to focus on the next 2 or 3 slides was published by GlaxoSmithKline from a Japanese group. And I would like to point out some examples from these publications on the next slide. What this group has done first is, in principle, to assess the possibility to use different PCR reagents for multiplex real-time PCR, comparing commonly used PCR reagents with the optimized reagent technology for multiplex PCR. On this slide, 2 different transcripts are shown, cyclophilin and fipronectin, which were analyzed in a duplex PCR reaction utilizing different starting amounts of total RNA. And as you can see in the lower panel of the figure, you observe what is commonly seen in non-optimized multiplex real-time PCR system, namely more shallow amplification curves, indicating lower PCR efficiency, and the lack of detecting low abundant transcripts or low expressed genes and low starting materials. However, this changes by using this optimized reagent for multiplex PCR, where you see high PCR efficiency by the slope of the amplification curves, and even small amounts of starting template or low expressed genes are equally well picked up also in a co-amplification reaction. This example shows an actual gene expression measurement system where this group investigated the effect of a certain drug compound on certain transcripts, in this case fibronectin 1, collagen type 1, and collagen type 3 transcripts, and comparing actual gene expression levels between singleplex PCR reactions and duplex PCR reactions, normalizing the transcript of interest to the normalizing reference gene in the same reaction well. And as you can see from the published results, the gene expression levels between single and duplex are always staying the same, showing that now reliable gene expression data can be also obtained in a multiplex real-time PCR environment. And this is also the conclusion which these authors have drawn from their analysis. They investigated 38 different gene expression assays, which all worked under universal amplification conditions without the need to further optimize these assays. And they came to the conclusion that with this new innovative reagent technology, they are now able to overcome the commonly known problems of duplex real-time PCR. They are just about to publish a new paper, as I have heard, which not only shows this for duplex real-time PCR analysis, but bringing this to a completely different level, where they were able to analyze in the same reagent environment, up to 4 transcripts per reaction. This is also of quite value in genotyping approaches. In this case, these were data which were kindly provided to us by David Whitcomb from DXS Limited, who was interested to investigate the copy number of the biomarker cytochrome P450, and in this case, particularly the 2D6 allele. The 2D6 allele is of particular importance since it's involved in many metabolism in the metabolism of many drug compounds, and it can be present in the genome either in a deleted version, in a duplicated version, or can consist or be present in multi-duplications. And this would then subsequently lead to reduced or otherwise increased metabolism of many drug compounds. So from this point of view, it's very important to assess the copy number of this particular allele, since this will lead to an effective and safe dosage of certain drug compounds for patients later on. And as you can imagine, a twofold difference in real-time PCR is not very easy to establish, but David Whitcomb could nicely demonstrate that even in a multiplex PCR environment, you can do a very accurate gene copy number determination for this biomarker with this new reagent technology. A newer technology, which we have just introduced 2 months ago, is the so-called QuantiFast reagent technology, which improves the throughput of real-time PCR, which can be often limited, particularly when biomarker programs are ramping up later in clinical development. And with this new reagent technology, you can save runtimes and increase your throughput up to nearly twofold for both one-step and two-step RT-PCR. And in this case, I just have picked out one example utilizing the ABI 7900 platform and talking about an 8 hours shift where you can in principle double the throughput of your experiments. And the nice feature on this new technology is that it's not only applicable to so-called fast real-time PCR cyclers, but that it can be used as well for standard real-time PCR instruments, which are not specifically adopted for fast cycling, and you can still increase your throughput by a factor of roughly 2. And all these reagent technologies combined provide very accurate and reliable gene expression results, as is shown in this comparison. In this model system, we have spiked known copy numbers of interleukin-8 transcript into a non-relevant RNA background in which this gene is not expressed. And then we have estimated the relative abundance of interleukin 8 normalized to GAPDH from various starting amounts of template and compared the results with the expected results, since we knew how many transcripts we have included in the various samples, which is represented by the thread line. And as you can see, with this improved reagent technologies, we get much more tight gene expression results, allowing for higher reliable and accurate gene expression results. This technology can be also adopted to faster analysis when doing screening directly from cell lysates from multi-well plates, for example. And this new approach eliminates RNA purification at all, leading still to accurate gene expression results. And let me briefly introduce this new technology, which is called FastLen cell RT-PCR, which is conducted in a one-step RT-PCR procedure, which really enables you to increase throughput in a very convenient manner. You start with the cell culture plate. In this case, we picked out a 96 well plate. You wash the cells for 1 minute, and then you lyse the cells, which takes about 5 minutes upon adding the lysis reagents, which not only lyses the cells, but also stabilizes the RNA, and in the same step also eliminates the background genomic DNA, which would otherwise contribute to the overall fluorescent signal of your analysis and would compromise the accuracy of your gene expression results. After this 5-minute incubation step, the sample is heat inactivated for 5 minutes at 75 degrees, and after roughly 15 minutes, all the samples from your 96 well plate are ready to go into real-time one-step RT-PCR analysis using either TaqMan probes, cybergreen analysis, or even multiplex approaches. And this slide shows the efficient removal of the genomic DNA so that you can be really sure that your gene expression signal is really derived only from the cDNA signal, which is indicated for different template amounts in this lower line. If the gDNA would be not removed from your sample, the fluorescent signal for the genomic DNA only would be indicated by this line. This can be easily tested in your real-time PCR setup by leaving out the RT enzyme from the cDNA synthesis, conducting the normal gene expression analysis, and the fluorescent signal which shows up in such so-called no RT or minus RT controls is only contributed by the genomic DNA present in the sample. But this genomic DNA can be very efficiently removed in this 5-minute short incubation step in the procedure, showing in this example a greatly reduced DNA amount which will not any longer interfere with the gene expression results. This procedure works very reliable and reproducible over the complete plate, in this case utilizing cybergreen detection for the analysis of transcript from a HeLa cells cultured in a 96 well plate, and you see that the coefficient of variation is pretty small for all the different wells. And the same holds true in this case in a multiplex PCR environment, where we have analyzed and normalized the NF-kappa-B expression to the GAPDH normalizing gene, co-amplified in the same reaction well with the new multiplex technology. And this can be done also with a high reliability and reproducibility directly from lysates for high throughput screening procedures. So this technology requires no RNA purification, provides significant time savings, and can be readily automated on liquid handling platforms. There are only 3 steps necessary to go to your real-time PCR, and most importantly also includes already genomic DNA removal step, so that the gene expression levels are true signals derived from your cDNA. This technology can be combined with either of our downstream real-time PCR reagents for cybergreen or TaqMan probe detection, and even for multiplex PCR, even further increasing your throughput. And these can be also combined with ready-to-use gene expression assays, for example, for cybergreen-based analysis, which I'm going to introduce on the next 2 slides. We offer on our website, or if you would like to take a shortcut, go directly to geneglobe.com. Ready-to-use real-time PCR assays for cybergreen analysis for all transcripts for human samples, rat samples, or mouse samples. And if you access our website and look for particular gene expression assays, you will see a graphical display which shows you the position of the PCR product on the transcript, which shows also polymorphic sites and relevant silencing RNAs, which can be also obtained from this website. And will introduce further information like the size of the PCR product, which exons become amplified, and also contains a direct link to the NCBI homepage from where you get specific information on the molecular and cellular background for the various transcripts of interest. We offer currently more than 180,000 validated QuantiTaq primer assays, which work all under one unique cycling protocol on all commercially available real-time PCR cyclers. And when using those in conjunction with the QuantiTaq Cyber Green kits, they can be used for both one-step and two-step real-time PCR. And as I have shown before, also for such screening purposes, such as direct analysis of gene expression data from the cell lysates, offering a very cost-saving and convenient procedure in contrast to more expensive TaqMan probes. One very hot topic in biomarker discovery and utilization are so-called microRNAs or small regulatory RNAs. And with this slide, I would just briefly like to introduce microRNAs. MicroRNAs are generated in the nucleus, exported to the cytosol, where they become processed by a molecule called dicer, and are subsequently incorporated into the so-called RISC complex. And these microRNAs can in principle act in two ways. They can act like the functional genomic tools, the silencing RNAs, by degrading the messenger RNA, or they can act as a translational repressor, preventing translation of the messenger RNA at the ribosomal site. Whether it acts in either of those ways, it's mainly dependent on the homology of the microRNA to its target transcript. But analyzing microRNAs or so-called small non-regulatory RNAs is really a challenge, and this is mainly due to two reasons. On one hand, as I said, these microRNAs are very short regulatory non-coding RNAs, pretty much in the range of 20 bases. And this makes clear that it's very complicated on one hand to efficiently reverse transcribe the microRNA, as well as to detect it into an amplification procedure, since its overall size is smaller than a regular amplification product. Another obstacle is that you would like, of course, to utilize one technology to reverse transcribe and detect all microRNAs to be sure that all are detected with the same efficiency and sensitivity, and that not the procedure itself introduces variability in sensitivity and efficiency of detection. Another problem arises from the sequence context of the microRNAs itself. MicroRNAs can be present in a family member context, and individual family members may differ by as little as one base, which is of course very hard to detect in a PCR-based approach. So the question is, how can one selectively amplify and detect different family members? And ideally, such a technology would be not only suitable to analyze microRNAs from your cDNA sample, but should be also applicable to analyze messenger RNA, so that you can analyze both target molecules of interest from one and the same cDNA sample. And ideally, such a technology should be also open and applicable for new short regulatory RNAs, which are currently in the process to be discovered, like, for example, SNO RNAs, which are other small regulatory RNAs. And this has been solved with a novel technology, which I would like to introduce on this slide. And I would like you to focus first on the right side of this schematic drawing, which represents a microRNA or other small non-coding RNAs. With a relatively simple trick, we extend the length of these microRNAs by an enzymatic polyadenylation of the microRNA in an enzymatic reaction. This gives rise to a microRNA molecule, which harbors, like messenger RNA, a poly-A tail. And this poly-A tail can now be used to utilize oligo-DT primer for cDNA synthesis, which in addition harbors also a universal text sequence. So this reverse transcription reaction, which can be utilized for all different microRNAs present in the sample, yields microRNA cDNAs with a poly-DT sequence carrying a universal text sequence, which can be utilized later on in the PCR step as a common downstream PCR primer. And the microRNA specific PCR primer is then used to amplify the microRNA. And as this procedure implies, this procedure and technology is applicable to all small RNAs equally present in the PCR, in the RNA sample. And it does also not interfere with the reverse transcription of the messenger RNA. And for this reason, you can use and analyze both microRNAs and messenger RNAs from the very same sample. In the subsequent PCR step, we make use of the QuantiTaq CyberGreen PCR master mix, which comes along with the MyScript universal primer, which binds to the poly-T tail of the microRNA cDNA. And on our GeneGlobe webpage, where you can purchase siRNAs as well as gene expression assays, we will have also all microRNA specific primer assays available, which can be combined in the real-time PCR assay to sensitively and efficiently quantify all different microRNAs which are currently known to date. And the same technology can be used also to analyze the messenger RNAs, which may be affected by the regulatory function of the microRNA. This slide addresses the other problem, namely the high sequence homology, and I would like you to focus on the microRNA LED7B and LED7B-C, which differ only by one base in the context. And through this stringent validation procedure and high specificity PCR reagents, we are able to design assays which to nearly 100% only detect in a CyberGreen-based detection approach the specific microRNA of interest, as is indicated here by the contribution of the fluorescent signal to the specific microRNA under investigation. So that shows, for example, that for the LED7B microRNA, there's only a contribution from the homologous LED7C microRNA differing only by one base of roughly 2% to the overall fluorescent signal, whereas the LED7B microRNA contributes to less than 1%. So really insignificant amounts, showing that highly specific and sensitive quantitation of specific microRNA family members can be achieved with this technology. With this slide, I would like to conclude the topic of microRNA or other small regulatory RNAs, showing the overall workflow, reducing greatly variability. So you start with a total RNA isolation, which can be done with a microRNA-easy kit, which isolates not only microRNAs, but also messenger RNAs and other small regulatory RNAs. But there's also one protocol included in this kit, which enables you also to selectively purify only small regulatory RNAs if you are interested in discovery programs, for example. Then you utilize the MyScript reverse transcription reagent to simultaneously reverse transcribe not only microRNAs, but also your messenger RNA. And in the subsequent RT-PCR step, you can utilize the various reagents and validated assays to quantify and sensitively detect your messenger RNAs of interest as well as your regulatory microRNAs. And this technology platform is open for new upcoming biomarkers such as other regulatory small RNAs like SNOME RNAs, since it can be, due to this unique outline of the technology principle, easily adapted to new biomarkers. So I hope that I have shown in the past slides that QIAGEN has much more to offer than only DNA or RNA sample preparation tools, but that we rather provide tools to standardize whole assay workflows from sample to assay, starting with collection devices, stabilizing the biomolecules, not only for RNA, but also of proteins, for example, Combining this with gold standard sample preparation technologies, which could be either done in a manual fashion or on different automated platforms, and also providing along the relevant assay reagents for example, mass spec analysis of proteins or real-time PCR analysis for gene expression results. And I would also like to point out that due to our possibilities in diagnostic assay development through our diagnostic branch, we are more than happy to provide our competence in partnership with pharma partners to develop companion diagnostic assays along with the drug compounds of interest. This is underlined by enabling automation tools which are available for purification of all different biomolecules like DNA, RNA, and protein, starting from various very diverse starting materials such as blood or tissue or saliva, and enabling automation not only for higher throughput but also for low to medium throughput demands. And we just recently introduced a novel instrument to the market which is called the QIAcube, which is in principle a fully automated centrifuge-based sample preparation tool, very cost-effective, and simply takes your well-known manual sample preparation protocols into a low to medium throughput automated system without any changes to the purification technology. But of course, we also provide high throughput automated solutions, which are also CFR 21 Part 11 compliant, definitely very important for your biomarker research programs. And in the partnership with Pathway Diagnostics, we can provide these technologies not only in your own lab, but through Pathway Diagnostics also through their services where they are involved in biomarker assay development services, as well as biomarker clinical validation services, making use of a number of QIAGEN's technologies. And as I said, QIAGEN would be more than happy to bring them your assays also as a companion diagnostic product in a partnership to the market along with your newly approved drug. So I think I would like to put a lot of emphasis that QIAGEN not only focuses on academic research market, which is still, of course, extremely important for us, since this is the market from which all the developments proceed, also to biotechnology and pharma companies. And our focus has been very much in the past years on addressing the rapid growth markets, such as the pharmaceutical market and molecular diagnostic markers. And nowadays, QIAGEN products are involved in more than 100 clinical trials, and especially also the stabilization products through our joint venture in pre-analytics, diminishing a lot of variability in your biomarker research programs, are nowadays also involved in more than 50 clinical trials. I would like to summarize this and point out this importance in one case study, namely our more than 10-year lasting partnership now with Merck. And this slide was presented in a joint presentation by QIAGEN and Merck on the Global Pharma R&D Summit at Anaheim last year in October, showing the very successful HPV vaccine program from Merck for the Gardasil vaccine, where QIAGEN was able to support Merck throughout all clinical phases with different throughput needs for their purification of different starting materials, providing automated solutions when coming to higher throughput, and also providing all the necessary real-time PCR reagents to conduct this genotyping effort throughout all different clinical phases. So I think a very successful partnership for both parties. With this very last slide, I would like to summarize my presentation. I hope I could demonstrate that the standardization of workflows is possible with a couple of new technology solutions which help to standardize workflows from sample to assay, utilizing, and what I believe is very important, same technologies throughout the different processes like discovery, preclinical development, going through clinical development, ensuring high reliability of the various methods, and making these technologies also applicable to automation as it's necessary to meet the different throughput demands. I have introduced several technologies to stabilize biomolecules in order to reduce variabilities, to increase sensitivity of different starting materials such as proteomics workflows by reducing sample complexity, and by immortalizing not only genomic DNA samples but also RNA samples for subsequent genotyping and gene expression analysis, providing not only sample preparation reagents for all these different tasks but also reliable assay reagents along with contents. And as I have pointed out earlier, we would be definitely happy to, in partnerships with our pharma partners, to develop companion diagnostic products through our diagnostic branch. We can offer, and I think QIAGEN is one of the unique companies positioned in the market, freedom to operate for all these different procedures. Our products are manufactured under validated processes which are GMP compliant and ISO compliant. And what may be of particular importance in biomarker programs when scaling up biomarker programs is to provide homogeneous lots accordingly certified according to the needs of our partners. We can do also lot reservation and lot tracking, so everything which is required in a highly standard operation procedures driven development and biomarker program. With this, I would like to thank you for your attention and would be happy to answer your questions. So one question is whether the presentation will be available later on. We will definitely archive this presentation and will make this accessible through our website to our customers, also if colleagues of yours may have missed the presentation. I see another question regarding to multiplex real-time PCR gene expression analysis and whether this is also feasible in a one-step RT-PCR environment, since this would greatly improve the overall throughput. Yes, this reagent technology is not only available as two-step RT-PCR solution, but also as one-step RT-PCR solution and can be equally well used also for this. I can also tell that for those who are interested in infectious disease programs, where the pathogen actually is the biomarker itself, we have recently introduced a new technology to the market, which is called the QIAplex technology based on the Genotyping platform, which enables simultaneous detection and amplification of actual panels of pathogens in one run. And if you are interested, our pharma key account managers can definitely give more information also on this technology. There is another question on stabilization of proteins through the All-Protect technology. As I have shown, we could demonstrate already preliminary results showing that enzyme activity is preserved by utilizing this technology. In this particular question, the question has been whether also phosphorylation of proteins will be maintained by this new technology. This is still under investigation, so we have no final conclusive data on this, but we will update our information on this as soon as it is possible. I would like again to point out that we have one technology available with which phosphorylated proteins can be selectively enriched, which should aid already in the biomarker development programs according to phosphorylated proteins. So if there are no more questions, I would like again to thank you very much for your attending the seminar and hope it was helpful information for you, and would like to welcome you back again to another QIAGEN online webinar. So thank you very much.